That means that, unfortunately, FDA currently sees this as a drug. And that's specifically when you're placing it in any tissue or inside any human. There's a little wrinkle to that if you use it topically, which we could get into. But they see it as a drug.
Welcome back, everybody, to another KOL Sessions podcast. Today we are here with Preston Wolfe, who's a PhD in translational orthopedics. We're going to be unlocking the healing power of exosomes. This is Exosomes 101. Preston, thank you so much for being here.
Thanks for having me.
Yeah, how was the trip over?
It was good. Had a little rain, delays out of Atlanta, but made it in time, so all good.
Nice. So we're a bit lucky today. He's actually going to be presenting on exosomes at the Toby conference starting tomorrow. But we're going to give you a little sneak peek here and get into some basic science and also talk a little bit about how exosomes can or maybe cannot right now translate into clinical practice.
Yeah, definitely a big topic. So hopefully just give a little bit of science behind it and maybe bust some myths and give some knowledge behind it as well.
Yeah, absolutely. So tell us a little bit about your background. You know, how did you get into exosomes and just into orthobiologics in general?
Yeah. So I did my PhD at the University of Missouri, where I mostly focused on diagnostics for hip dysplasia on dogs and humans, and really try to understand the pathophysiology of it down from a cellular, molecular level all the way to a tissue level and biomechanics. Exosomes were a new topic when I was in grad school, and I mean, it seems like it's gone quick over the past couple of years, 3-4 years, but it was a fringe topic at start. Not many people knew what they were, and when I was starting industry, it became more and more popular in the orthopedic space, so just kind of started diving into it a little bit.
So it seems it's such a cool word, exosomes, right? It's very trendy, and I've seen a lot of people get exosomes treatments and they have no idea what they're getting. So can you tell us, exactly what are exosomes and kind of where do they come from? Give us, the scoop on it.
Yeah, so exosomes is a specific size of an extracellular vesicle. There's a few different ranges out there, but typically between 50 and 200 nanometers in size is what an exosome is. And how it's produced is the cells take in signal and react to the signal by forming multivesicular bodies, MVBs, and then it releases those out of the cell to respond to the environment however it sees fit. Sometimes that's good, sometimes it's bad. I mean, we talk about it in cancer a little bit when we're trying to diagnose cancer, and there's thoughts as well that cancer cells release these exosomes or extracellular vesicles, and that's what causes recurrence. So sometimes they could be really good depending on the cell source, honestly, and how they're reacting to their environment and what their goal is as a cell.
So it's essentially the way that a cell communicates to another cell, right?
Absolutely, and modulates its environment.
Okay, and can any cell produce exosomes?
Yeah, I mean, even milk, urine, everything. There's exosomes everywhere.
Everywhere.
Yeah.
So in layman's term, it's basically a text message, right? So if I want to communicate to you, I write this message in a little bubble and I send it over to you, you receive it, and then you do something with that message.
Yeah, that's a very good way to put it. Yeah.
Cool. So last time I checked, I did this lecture, maybe it's already been a few years, but there was around like 10,000 identified exosomes. Is there more now? How many exosomes are there?
And I guess, what do you mean by that? How many are there?
Like the ones that we can actually identify as like an individual exosome or label it in a certain way.
Yeah, I mean, they're per milliliter, say, in your serum, in your plasma, there can be millions, billions of exosomes.
Yeah.
We don't really know how much we need for a therapeutic effect, but I don't think there's really a specific number on it. There's different types of exosomes from different types of cells. So say a synovial cell versus a cartilage cell, they release different types of exosomes.
Okay.
So it's really tissue and cellular specific.
Got it. Can you walk us how an exosome product is, or maybe not a product, but just an exosome in general? Like what's the biogenesis?
Yeah. So it takes in that signal from its environment, and whatever the cell is, it responds in a certain way and creates those multivesicular bodies that I described earlier, and then they bind to the plasma, the cytoplasma, and release its contents to the environment.
And why are lipid bilayers and CD markers, like CD9, CD63, why are they important in identifying and characterizing exosomes?
Yeah, so the lipid bilayer obviously gives it its structure and its shape, and so it's just like a lipid bilayer in a cell. There's not really anything specific about the lipid bilayer, but that's where everything kind of lives that determines if it's an exosome versus a apoptotic body. So after a cell is dying, it releases apoptotic bodies. That's what really differentiates it, those CD markers. There's other markers on the outer portion of it that allow it to bind to other cells or have its interaction or its proposed mechanism of action, really. And there's three ones that we consider from exosomes: CD9, CD63, CD81.
Okay.
We look at those three. Usually we want to see a high level, high concentration and purity of two of those to consider them exosomes.
Okay.
And you can do that through various different tagging and analysis methods.
Can you go a little bit into that since we're diving deep? How do you how do you tag an exosome, right?
So you can test it two different ways. You could do a typical Western blot and really take those exosomes and break it down and then just look for those proteins. If you want it on an intact vesicle, you can do it pretty much two different ways. You can do electron microscopy and do some type of, say, antibody gold labeling. One of the more common ways now is with nanotracking analysis. So nanotracking analysis lets you determine the size and the count, but just because you have something of a certain size and a certain count doesn't mean that it's actually an exosome. You also have to have the surface markers as well. So there's specific machines. Probably the best one out there is ZetaView right now that allows you to do the size, the count, and then also tag multiple different protein markers on the outside. So you put an antibody that has a fluorescent tag on it in a certain wavelength. You do three different ones so you can detect them all at the same time. Then you use three different wavelengths of the laser with this NTA device to count and quantify the purity of the specific CD markers on the exosomes.
Got it. Amazing. How does exosome compare to a mesenchymal stem cell?
Yeah, so mesenchymal stem cells obviously were popularized by Dr. Kaplan, really the godfather of MSCs, and rest in peace, he unfortunately passed away recently. But he did a lot of great work in the stem cell realm. If you start to talk to the really basic scientists in this realm, they've stopped calling them stem cells because stem cells were pretty much meaning that they had the ability to engraft in, and then they had stemness, so they had the ability to differentiate into different types of actual cells that are differentiated. And so we think of chondrocytes, we think of ligaments, we think of fat, and we think of bone typically with stem cells.
Sure.
The multipotent stem cells. So because that was really what we saw in the laboratory, it wasn't really potentially true when you're talking about allogenic stem cells. We were seeing with different studies of tracking these cells after they've been injected in that they don't really, not many engraft in when you say inject it into a joint. And so, but it still had an effect. It still had a great effect on reducing inflammation and potentially having some type of proliferation of the cells that were already there. So Kaplan, not many scientists I feel like do this, but he backtracked and said, hey, maybe we need to rename this a little bit and not call them stem cells because that's what we see in the laboratory. Now, when we actually put them in human, let's call them stromal cells because they have an effect. They're from the stroma, the MSCs are, and we could still call them MSCs, but then also the analysis of these vesicular bodies that mostly are called exosomes that we first thought are trash. We realized that they were actually, we thought they were trash at first, that the cell just kind of pooping out its contents it didn't need, which is funny to think of now with all the clinical trials going on with them. But he said, well, this is a signal. They're signaling. The MSCs are really a really immature cell, but they have a lot of potential to influence their environment. It's probably through the signaling, probably more what we call the secretome, so everything that's being secreted by the cell, not just vesicular bodies or extracellular vesicles, but also various types of proteins. And so he started calling them MSCs as signaling cells or stromal cells.
Got it.
Hope that answered your question.
It does, yeah. So it sounds like, again, just bringing it down, the mesenchymal stem cell, now kind of known as a mesenchymal signaling cell, the way that it signals is via the exosome.
Yes,
Is that
one
accurate?
of the ways.
It's
yes.
one of the ways.
Yes.
Got it. Okay. So I guess the question I was trying to get to earlier is, how do we know how the cell makes the exosome? How do we do it in practice, right? How do we collect them, and how do we create an exosome product?
Yeah, it's a very good question. Typically, they're grown out in a laboratory and they're kept to a certain passage, so they haven't differentiated into more fibrous cells. Mm-hmm. So we typically keep them at a low passage, and then everything that's released to that media is what we call the secretome.
Okay.
So everything the cell is secreted over a couple days. You take that media, you isolate it down through various techniques. Depending on the technique, depends on how pure of a product you get, and that's why. Well, before I get into that, it determines how pure a product you get. So you could do ultracentrifugation, which is non-specific, but you get a high return on extracellular vesicles because it's not so specific. There's filtration, which is a little more specific, so you get less of a return from what's in the soup the cells released. And then you could do like immunoprecipitation, which is very specific because you're tagging specific cell surface markers, potentially washing out all the rest of the contents, releasing a really pure product for, say, if you're trying to get specifically exosomes with a certain cell surface marker, but you get a lower count out in the end. So depending on the way you do it, probably will depend on if you call it an exosome product or if you call it an extracellular vesicle product. Because the less specific you are, there's other vesicles there, like apoptotic bodies, ectosomes, another term as well, but they're various different sizes.
And what's the actual host? What's the source? Is it the donor gives blood, bone marrow, adipose? Where are they actually coming from?
Yeah, I think it depends on the company, really.
Okay.
There's some that will take a patient's own plasma.
Okay.
Own blood and isolate them from platelet-poor plasma, say. There's other ones where they'll do other autologous cells like bone marrow or take the PPP from bone marrow aspirate. What we're seeing a lot in the clinical trials is more birth tissue MSCs, growing those out in the laboratory either from umbilical cord or placenta, and then taking the exosomes from that or extracellular vesicles from that. That's what we're seeing in most of the higher level clinical trials that are more rigorous.
Now, is it, my understanding is, for instance, if you want to get mesenchymal stem cells, right, I can grab birth tissue or bone marrow, extract the cell, and then I pass it down and I start to, expand it and multiply it.
Yes.
Is exosomes the same thing, or when I get the donor, there's already a finite number of exosomes and I'm just extracting them from the source?
The source will be producing them over time.
Okay.
And why they, in these more rigorous scientific studies, why they grow them out to a certain passage and put certain things on them instead of just grabbing them straight from the initial cell and let it passage a few times, is they want to be very specific on how they do things each time to have potential more consistency of what the exosome is and more consistent of what the exosome would do. Now, if you just pull it from, say, your blood versus my blood, for example, they're going to be very different. Mm-hmm. So I think it depends on how you're doing it, but yeah, they have certain conditions in the laboratory setting to try to keep it as consistent as they can. Lot variability, different things like that.
Gotcha. Now, obviously, like you mentioned, there's tons of these exosomes, right? Do we know, have we identified a specific one with a certain marker that does an anti-inflammatory effect, one that has a proliferative effect? Have we been able to identify specific markers that do certain things?
There are some people out there that are trying to specifically point to one cell surface marker. CD73 is one some scientists think are really potentially beneficial. That's specifically because it is really a surface marker that can break down ATP, ADP, or AMP and move it down that pathway. So then you have adenosine and you have phosphate. Mm-hmm. Because when you have ATP outside the cell, it's a signal to the body that, hey, cells are dying. We shouldn't have ATP outside the cell. It has no purpose.
Sure.
So if that's outside the cell, which could be happening in damaged areas, if you inject, say, exosomes that have a specific CD73 marker, the thought is that you push it down and separate those adenosines from the phosphate, and it's more anti-inflammatory than it was before. So CD73 is one that specifically people think are anti-inflammatory. There's a few other ones that we believe may have an effect on making monocytes in an area or M1 macrophages into M2, and then it could be immune modulatory and anti-inflammatory. On the proliferative side, I don't believe we have anything specific from the cell surface marker, but some people are claiming that the inner content, so like say microRNA, is what would be entering the cells in the area that are damaged or inflamed and potentially guiding them to translate proteins that are then pro-proliferative again. But I think specific ones that have a specific effect are more on the inflammatory side and more on the immune modulatory side.
Got it. Now, what are your thoughts on the collecting or trying to create an exosome product versus via density gradient?
Yeah, so density gradient's an interesting one because that's probably a little more cost effective than having maybe a laboratory set up with filtration or having $100,000 ultracentrifuge that you have to do for hours at 100,000s of Gs. So it's intriguing because you could potentially do it in a centrifuge and a basic centrifuge and really pull it out of whatever fluid you have, but it's very non-specific. I would say it's probably the most non-specific, so you're probably getting other type of conglomerates of lipids or apoptotic bodies as well. I've seen some papers out there that have been doing short centrifugation times with a PEG or dextrin density gradient, and even at 5 to 10,000 G, I believe it was, of centrifugation, they did transmission electron microscopy, and they really didn't have a great count. So so maybe it's not as great as we think. I also have a worry that there's potential some type of reaction you could get if you don't remove that PEG and dextrin before placing into a human. Dextrin is obviously just a sugar, but PEG is polyethylene glycol. It's something that kind of came up, especially with recent vaccinations during the pandemic. People have sensitivity to PEG, polyethylene glycol. So I think injecting that into a place that has sensitive tissue, like a joint, may have more issues than just, say, intramuscular, but I don't have any data to back it up.
Gotcha. And how can this word exosome be problematic from a regulatory or scientific perspective?
Yeah. So as I mentioned earlier, there's a lot of different vesicles of different sizes. They have different potential effects. Some could be beneficial, some could be inhibitory. And with the current techniques outside of immune precipitation, which you won't get a huge concentration of, you're really getting a kind of pool of vesicles, not just one type of vesicle, just the exosome. So when you go to, say, a regulatory body and they say, you want to say you have an exosome product, and then they want to test it, and they're like, well, there's a lot of other things there.
Here,
how are you sure it's the exosome that's having the effect? That's where it potentially gets a little hairy.
So it's a bit of an umbrella term.
It is. So that's why we see more of ISEV, International Society of Extracellular Vesicles. They really caution against using exosome. If you do think you have an exosome product of a certain size range, call them small extracellular vesicles. And we're seeing that in clinical trials as well. People more going for that term. It's a little more, FDA seems a little more comfortable with that term, small extracellular vesicles than exosomes.
Got it. Can you compare this to how PRP, we've been classifying it as, and again, this has also been changing, right?
Absolutely.
But low dose, high dose PRP, leukocyte poor, leukocyte rich, now monocyte rich, neutrophil reduced. How do you compare that landscape?
Yeah, if you have isolation techniques that are non-specific, you're obviously not just getting exosomes. So if you say, for example, leukocyte rich, well, are you talking about the lymphocytes? Are you talking about the neutrophils? Talking about the monocytes? What are you talking about here? And I think we're getting more granular on that on the PRP side of maybe talking about monocytes more, or talking about neutrophils more, and trying to understand what lymphocytes do a little bit. So I think it's kind of comparable. I think it's also, if you were to kind of take a PRP product and you market it as, I have an autologous PDGF product. Well, you have more than just PDGF there.
Yeah.
That might be the important one, or it could be VEGF, and
Sure.
you're certainly going to have it there.
Yeah.
And with current isolation methods, you're going to have exosomes there.
Yeah.
But it's not just that specific thing. I think we have to be a little more, unfortunately, broad with the current techniques we have.
Sure. So is it safe to say that PRP has exosomes?
Yes.
Right?
Yeah, there's several studies that have shown that.
They've shown that. Okay.
Some people claim that might be a very important
How
part
they
of it
work.
as well. Yeah,
Yeah.
I think the
Or
growth
one of the
factors do a
pathways.
lot.
Sure.
But obviously, the platelets, whatever they release,
Right.
is a lot. And the platelets can release exosomes.
So when you get a PRP treatment or a bone marrow concentrate treatment, you're also kind of technically getting some sort of exosome treatment with
Yeah.
it.
Yeah,
Got it.
just not purified or non-specific.
Non-specific.
We don't really know the concentration. Depends on a lot of things.
Sure. All right. Where do exosomes currently stand with the FDA? Because this is it's awesome to talk about. I think exosomes have a lot of promise. But, there is the truth of the matter is the regulatory landscape, at least in the United States. Where do they stand, and what does it mean that they're regulated under Section 351?
Yeah, that means that, unfortunately, FDA currently sees this as a drug. Mm-hmm. And that's specifically when you're placing it in any tissue or inside any human. There's a little wrinkle to that if you use it topically, which we could get into. But they see it as a drug, and it's a tough area. I think it's the same thing, the same way they treat kind of SVF.
Yeah.
If you're isolating something specifically outside of what naturally is coming out, they see that as a drug.
More than minimal manipulation, in a sense.
Yeah, I think that's a interesting thing to dive into, because is centrifugation or filtration, is that really minimal manipulation?
Yeah.
So I think it's more where we saw exosomes coming from at first. It was all allograft. No one was talking about autologous. So I think that kind of maybe jaded them a little bit to even talk about autologous, because I haven't seen them come out with any type of definition or any type of line in the sand of, well, what if I'm getting the exosomes from PRP or from bone marrow aspirate or from microfragment adipose? They haven't drawn a line on that to say if it's a drug. So it'd be great if someone could ask them that question and get some clarity.
And kind of not to go off topic, but we always assume that the source is going to be human blood or human tissue. Are people extracting exosomes from animals and trying to put that into humans?
In the U.S., not so much, but in Europe, definitely on the aesthetics cosmetic side. There is one in the U.S. that's doing bovine umbilical or placenta, but that's all for cosmetic use. But I mean, the cosmetic space in Europe is all over the place. I mean, even plants, pumpkins, for example, is one. So yeah, they've been extracting from other places. In the U.S., what we see people kind of marketing on the orthopedic side is more either from PRP, from plasma, or from these human birth tissues.
Well, I guess that's a good segue for the next question. It does seem, especially in the U.S., where in an orthopedic side, we're very, I don't personally use exosomes because, FDA regulations, but on the aesthetic sides, I've been seeing them been used all over, and it's like candy, right? So what is the workaround, and how do the rules apply for cosmetic or topical uses of exosomes?
Yeah, so that's not under the FDA. That's under the FD&C.
Okay.
The MOCRA Act specifically is what kind of defined this and allowed it to be used. So they're able to get away from it or with it on the aesthetics cosmetic side as long as they use it topically. And they can't make claims about pathology, so they can't make claims about any specific kind of topical. I'm not a dermatologist by any means, but any type of topical disorder or disease, they can't make any claims on that. They can just use it topically and say for beautification or hydration. I think it even is potentially a gray area if you could even say increases collagen production.
Okay. And
you can't use it in microneedling or anything. It has to be specific topically.
Got it. And the other place I've seen it is IV exosomes. That I would say that's a no-go.
Absolutely.
Okay, got it. What about physicians that say, well, I'm doing a study, I got an IND, can you do it that way? What's the IND pathway like? Can you use exosomes through that?
So you can only use it, an IND product for the IND study. So if someone comes to you and says, hey, we have clearance from the FDA to use this as an IND, Well, if you're not part of the study in one of the sites, you can't
Right.
use it. That's not considered off-label use because they're going for their on-label indication.
Got it.
So there's no technical on-label indication yet, so you can't even use it off-label or claim that.
Got it. So what that means, if I'm a physician and a company comes to me and goes, hey, we have this exosome product, we have an IND, we're doing a study on it, go ahead and use it.
Yeah.
But if I'm not part of the study, I'm not part of the IND, I'm out of bounds.
Exactly.
Okay.
Exactly correct.
So that's a good point, I think, to
Yeah.
understand, right?
Yeah.
All right. So because of these regulatory issues, there have been multiple warning letters.
Mm-hmm.
What are your thoughts on that part of it?
Yeah, the warning letters have typically gone to the manufacturer.
Okay.
And they typically try to get them for any type of claim they've made. FDA's even started looking at social media, which is interesting.
Yeah.
I think they recently cited an Instagram post, which was a first, I think, of its kind. So they're finally coming around to social media.
Yeah, they're looking.
But typically it was just what was on people's websites.
Yeah.
And it's typically just on the manufacturing side. You kind of think of it as illegal drugs. They don't really go after the user. The FBI doesn't usually go after the user. They go after the manufacturer and distributor.
Sure.
So it's the same thing we see on the exosome side. We see them going after the manufacturer. They typically get them for manufacturing practices. A lot of times they'll say they're GMP, but they don't have any third party, either through the FDA, through the AABB, which is blood banking, or AATB, which is tissue banking. They typically don't have those kind of clearances or those third party oversight to ensure that they have GMP. So they usually go after them. And if you read through the documents, it's usually multiple infractions on the GMP side, which are old. The GMP kind of factors are old, but that's what they always hit them for.
Okay. Well, shifting a little bit to, I guess, clinical applications. In orthopedics, what conditions or injuries do you see as most promising? We'll start there, and then maybe if we know the mechanism of action and how these could potentially be helpful in orthopedics.
Yeah, so all the clinical trials on the orthopedic side that I've seen currently are for knee osteoarthritis.
Knee OA.
There's specifically 2 active ones in clinicaltrials.gov right now. There may be more ramping up, but we typically see knee OA as the primary.
And it's interesting, a lot of the studies that have been done are is it in respiratory disorders?
Yeah.
And why do you think that is?
Yeah, it was a side effect of the pandemic as well. They saw these as anti-inflammatory, and they could get clinical trials approved for this pandemic a lot quicker. So acute respiratory distress syndrome related to COVID, and it was usually inhaled. Some people do IV because the blood obviously goes through the lungs. At one of the first stops.
Sure.
So the thought was that they may be stopping, having an effect there. But it's really due to COVID is why we saw so many clinical trials on respiratory distress syndrome.
And I guess this is where I'm having the issue with exosomes, not to beat the drum too much, but it's kind of like, okay, we're giving these people these exosomes, right?
Yeah.
There's thousands, millions, billions of them. We're not really sure, which one are the good ones or maybe the bad ones. We're just kind of pooling them together and just handing them out.
Yeah.
What is the danger behind something like that?
Yeah, if someone's manufacturing them properly, they're removing all the potential immune components, and they've been doing the potency testing as well. I think that's an important key for the clinical trials, and the people that are doing it right that aren't.
Yeah.
The differentiator is having a potency test. So they understand that whatever their mechanism of action is, they're having this effect on it. And they've typically done preclinical trials already in animals and shown that there's no type of tumor growth or any type of issues with liver or lung toxicity. So when you're going to the people that have the right data, I think it's shown to be safe. Is it effective? I think time will tell.
Sure.
But the safety side has shown to be positive on my side.
And I guess those are the concerns, right? One is the lab doesn't have good practices. We've seen that with birth tissue products causing a lot of infections, right? Absolutely. That would be a big concern. The other concern would be that the text message that I send is to proliferate the wrong tissue, right? Or if someone already has an early cancer, that signal I'm injecting, makes that cancer grow more.
Exactly.
Are those really the key concerns?
Yes, those are the key concerns. I think on the birth tissue side, they've shown, I don't think they've tested it on anything that already had a previous tumor in remission, so I'm not sure if it's going to cause a reoccurrence.
Okay.
I don't believe they've tested that specifically, so if a product does come to market, it's probably going to be very specific on that case.
Sure.
Or maybe a contraindication. But yeah, if it's from the right cell and grown the right way, it's been shown to be safe in a population that only has one specific type of orthopedic condition, from what I've seen.
Now, you talked a bit about potency, right?
Mm-hmm.
For instance, platelet dose, white blood cell, it's just a simple cell count.
Yeah.
What are the methods to testing the potency of an exosome product?
Yeah, probably the one that's most advanced right now, they're doing macrophage polarization. So they have good data showing M1 goes to M2, and it can have that effect. And that's what they're showing as their kind of release criteria and their potency right now in one of the current clinical trials. I think that's what most people are going after. Obviously, there's a group that's doing the CD73, so they're probably placing ADP or AMP and showing it pushes it down to adenosine and phosphate, and they're showing it's having the effect at a certain level that they expect, and it's consistent. And that's typically the potency that they're going after. Yeah, they're not specifically going to right now placing anything like with chondrocytes and showing that it causes proliferation. I think there's a lot of variability with that, and they're not taking that risk. They're doing something more specific and outlined already.
Got it. Can you talk a little bit about that? That concept of M1 to M2 polarization.
Yeah.
Why is that important? That's kind of been big for us in terms of, for me, why am I switching from like leukocyte poor to a monocyte rich PRP?
Yeah.
Can you expand on that a bit?
Yeah, so monocytes are the one type of white blood cell that's circulating throughout your blood, but once it goes into tissue, depending on the environment and different things going on, it will become a macrophage. There's two different types of macrophages. M1 macrophage, which is pro-inflammatory, and M2, which is anti-inflammatory. So once it gets into an environment, if it's already an inflamed environment and maybe a chronic inflamed environment, it is likely that it's always going to go towards M1 because it's kind of just a feed forward at that point. But if you can potentially modulate that and move it towards an M2, you could change that environment and change the potential inflammation on the tissue that's around.
Okay. Yeah, that's super important, and I think a really hot topic right now, something that we're going to be learning a lot more about in the upcoming years. When I look ahead, I think we're going to know a lot more about monocytes.
Absolutely.
And potentially why we feel that bone marrow concentrate, right, sometimes works better than PRP, because it's just got much higher volume of monocytes. Would you think that's accurate?
Absolutely. I mean, the amount of cells, MSCs, that are in bone marrow is a fraction of a percent.
Yeah.
So is it actually the MSCs that are having the effect from the bone marrow when you're placing it into a patient? Some people like Dr. Fortier, who did a lot of research and did kind of the initial BMC validation for a product on the market, she's also stated, well, we know there's a high IL-1RA with BMC, even compared to their IRAP product in the veterinary side. And so she kind of thinks maybe it's potentially more of the monocytes, it's more of the monocytes than the MSCs, or it's kind of together, right?
Sure.
Synergistic.
Now, we talked a little bit about how you make the product. Then how do you preserve the product? We looked at some samples when we were testing birth tissue-derived products, and cryopreservation was one of the ways that they kept the product. And then we felt that if you don't do that process right, you can really kill a lot of the cells. So what does cryopreservation mean in the exosome world? How are these companies keeping these products shelf stable, and what does that mean?
Yeah, so the nice thing about the vesicles, the extracellular vesicles or exosomes, is that they don't have all of that machinery inside like a typical cell does. So you're not really trying to maintain that function. You're really trying to just maintain the structure and the contents that are already there.
Okay.
So the thought is, because you don't have the cellular function that you're trying to preserve, that cryopreservation really isn't as big of an issue as it is for cells. Because cells, you want to keep them active, and a lot of people use a DMSO to keep the cells from being fractured as much because they're much larger than extracellular vesicles. And so the DMSO is actually cytotoxic as well.
Yeah.
If you don't wash it out properly, then you could have cells that are just dying. Right?
Sure.
Now with these extracellular vesicles, they're much smaller, so they're less likely to get kind of fractured by that freezing process. And you're not trying to maintain that cellular activity on the inside. You're just trying to maintain either the cell surface markers or the microRNA on the inside, which if you're not going through multiple freeze thaws really isn't an issue.
Got it.
There is early data that you could potentially lyophilize these, so they could be shelf stable and you don't even have to keep it in a freezer, which would be, I think, huge. I think it'd be easier for you than having a negative 80 cc, 80 degrees Celsius freezer. Yeah. So there's good data that's showing the lyophilization may be possible as well. And then you just hydrate it.
How important is biodistribution and clearance when determining the therapeutic potential of an exosome?
Yeah, so if you're injecting into, say, a joint, you want to make sure that it stays in that joint for a certain period of time, for a couple days or a couple hours, depending on what you're trying to do. But then you also want to make sure that after it gets removed from the joint, say if it's not utilized in the joint or its byproducts aren't utilized in the joint, that it's going into the blood and going to be distributed through the lungs, the brain, the liver, the kidneys, and you want to make sure that it's not probably sitting there for too long or cytotoxic. And so it's really important when you're looking at the animal studies. That's one of the main reasons of the animal studies is you tag them, you see where they go, you see how long they stay, and then you do your necropsy afterwards and do all your histology slides and make sure you're not having an effect on these organs that you're not treating specifically.
Got it. Now, you mentioned a lot of the current orthopedic trials are in knee.
Mm-hmm.
You know, do you see this as a product that's going to be more of an immunomodulator in terms of reducing inflammation, improving pain and function, or do you see it as something that could potentially regrow cartilage or you know, kind of reverse the arthritic process? Yeah,
the first in human paper that came out, it was out of a group out of Chile with Dr. Maroon Curry, and they obviously was just the first in human to make sure it's fine and safe after they did all their animal studies. They did MRI analysis. They also did pain function scores. Pain function scores were great to a year. But they did an MRI analysis as well and did quantitative volumetric MRI of the cartilage. Obviously just one patient, but they saw that there was more cartilage volume.
Interesting.
I think that's promising, but then it's also how do you convince the FDA that it's a DMOD? There is no DMODs on the human market, so disease modifying osteoarthritis drug. And I think it's kind of teaching the FDA and having those conversations of, hey, this is actually regrowing the cartilage, and then that actually correlates to function. Because you could regrow cartilage, but you could maybe not have more function or less pain. As you know from your practice, you'll see patients that have very little or no degeneration, but they're in a lot of pain, maybe from a bone marrow edema.
Sure.
So I think having that conversation, really outlining with the FDA on determining, having a definition of DMOD is important, and make sure it correlates to pain and function, and making sure they go together. But to answer your question, it could have some cartilage regrowth potential, but again, early on, but we pretty sure that's going to have an immune modulatory effect.
So the early signs or the early data seems promising for orthopedic uses.
It does, yes.
Do you feel that, and again, I'm not going to hold you to this, but if I do a trial of, PRP and an exosome product, how much different is the clinical benefit going to be, or are we just, it's going to be essentially kind of the same type of scenario?
Yeah, I think that's really hard because they, in my mind, both will have an effect. Mm-hmm. I think the kind of promise from the extracellular vesicle or exosome side is it can be very consistent. You can have a consistent product every single time off the shelf. Probably the patient population you have in isn't the healthiest all the time. And you probably think, maybe these platelets aren't even releasing anything at a high volume of whatever growth factor of interest. So I think it's the consistency which is really intriguing, where the PRP, you could have an old patient that's 85 and may have not as robust of an effect as someone 25. So I think the consistency is what's intriguing about exosomes or extracellular vesicles compared to PRP. But from my perspective, there's been no comparison at this point.
Yeah, I think that's the big promise of allografts, right? Is the fact that you can if you can find the right dose of the right thing, and then you can get consistent, the same, there's no variability within the lot, every sample is the same. And oh, by the way, I don't even have to take anything from the patient, I just have it, like as another medication. It would be amazing.
Yeah.
And to your point, there are patients that you're like, hey, you're uncontrolled diabetic, you're on all these medications, like how good is your plasma, right? How good is your bone marrow? Probably not great.
Yeah.
So they would really benefit from, an allograft. I think that's the big promise, right?
Yeah, and then I think once you get to a really consistent product, that's when we can start to have a lot more scientific fun, and then you could have more of potential markers or something to look to before you even treat the patient and say, based off of whatever probably AI algorithm in the future, we know that this is probably going to be really effective for your type of osteoarthritis. But I think we have to get to that really consistent product before we get to that phase. People have tried it with PRP, but I think there's inconsistencies on both sides of it. And I think the AI side of things are really going to help build out that kind of prognostic before you treat the patient and determine what's best.
Why do you think it's so difficult? And I'm going to give you a little news flash here. We just got accepted for publication on Cures, a paper where we compare growth factor levels on PRP versus like Wharton's jelly.
Okay.
I want to say we had one exosome product and then other birth tissue derived products.
Yeah.
And what we saw was that in general, these birth tissue derived products had similar growth factor contents that PRP did, but there was just a lot of variability even within the lot.
Yeah.
So why is it so difficult to get these allografts to get standardized and that pretty much every product has the same level of things?
Yeah, I mean, you're taking it from various different donors. You're taking typically placenta or umbilical cord from various different donors over different times, and that mother or her pregnancy and that infant, depending on what she was potentially exposed to, could probably affect that quality. Now, I think the interesting thing about the extracellular vesicles from the birth tissue is that you can take a small amount of cells and obviously expand it, and you could probably use those multiple times and use that specific patient on maybe hundreds of thousands of doses. So I think that's what kind of leads to consistency of that product. But yeah, I think there's just so many variabilities even with the birth tissue, and maybe how it was processed a little bit. There could be small little differences as well.
Okay. Now, if you guys don't know, Preston knows everything about Missouri. Missouri has more than 6,000 caves. Is that accurate?
Yeah,
that's
crazy.
Last
I checked. Might have a few more now.
So what's the hidden cave of exosome science that we still need to explore?
Yeah, I think the hidden cave is really that mechanism of action, really determining what the effect is. I was at the ICT conference. I was fortunate enough to sit in on the signature series for extracellular vesicles, exosomes, and I mean, it was debates all day long about various parts of it. And I think there was one portion where it was an hour and a half on mechanism of action, and probably the top five of the top 10 exosome EV scientists in the world, all there sitting on stage. And they went over time, and I don't think there was a consensus on what the mechanism of action is. So I think that's kind of the hidden part that we're really trying to figure out, and everyone's trying to figure out, from the scientists to the physicians, everyone.
Let's say I'm a clinician and I get approached by a company selling an exosome product. We are in August of 2025 because things change and they change
very
rapidly.
Quickly. Yeah.
So, in today's market,
Yeah.
What advice would you give them?
Yeah, I would say first ask for their FDA letter saying that they can use this. And if it's an IND, that's not enough if you're not part of that study. But if you don't really care about that as a physician, you just want to make sure you're giving the patient the proper product and you're not really worried about the regulation, you're willing to take that risk, then I would say ask for their either NTA or TEM, either electron microscopy or nanotracking analysis, and ensure that you're getting the product in concentration of a certain size that you're looking for, and then ask for their cell surface marker data, and then ask if they have any potential mechanisms of action and they have any potency assays, release criteria for the lot you're getting. Because the last thing you want to do is think you're getting a product and then realize it's not potent at all, or you're not getting the dose you expect, or maybe they're all apoptotic bodies and they're not actually extracellular vesicles. Yeah,
I think that's super important. And a lot of times when the reps coming into your office, they know a little bit about the science, but once you start to dive deep, it starts, if they can't answer those questions or they can't show you the actual facts, that's where you have to.
Or they don't have a product manager to reach out to get that data. I mean,
Sure.
I wouldn't expect most sales reps to really have that data in their back pocket because they probably don't get asked for it much.
Sure.
But they should have the contact and they should have it stored away at their whoever they're working for and have those printouts. You think about it for a birth tissue product, right? Or a soft tissue product for a ACL reconstruction or repair. If you want to have the release criteria on your making sure there's no infections there or anything like that, you can ask for it and they'll give it to you. They'll happily give it to you, right? If you're doing osteochondral allograft, they'll happily give you that biocompatibility data and different things. So I think you should keep them to the same standard on these types of products because it is an allograft product, likely from tissue.
Got it. All right, so this has been awesome. By the way, I'm learning so much just talking to you, man. We're gonna do a little fun close here.
Right.
So, if exosomes were a superhero, what would be their superpower?
Yeah, that's a tough one. If they had a superpower, I think they could kind of read people's minds, but then also shift it, kind of like from Star Wars.
Oh, nice.
These
like
aren't
the
the
Jedi
droids.
trick?
Yeah, these aren't the
Nice.
droids you're looking for. So I think it would be that type of. Maybe they'd be Jedis. There you go.
Jedis, I love it.
Yeah.
All right, so translational orthopedics. What is harder, explaining exosomes to a doctor or explaining what you do at a cocktail party?
Yeah, I've got really good at the second portion of that. I'm still trying to figure out how to explain exosomes. I think everyone is, honestly.
Sure.
But I've found ways to, in Uber rides from airports to places like this, explaining pretty specifically what I do. And I think most people are familiar with PRP and BMC at this point, that it's been easier to talk about. Funny story, when I was in grad school, I was doing biomechanics research on a robot. A lot of my family's really smart, but they thought I was doing robotic surgery for a while. And I was like, no, that's not what I'm doing. So yeah, but I think the PRP, BMC side, people are really understanding it. Yeah.
All right, so Missouri is known as the show me state.
Yeah.
What's been your biggest show me moment in science?
Yeah, I think there's quite a few because I've got to be a part and see a lot of cool science over the years. I think on the orthopedic side, like kind of what I do now, I think the IO stuff, when I was coming out of graduate school, I didn't really have a lot of exposure to the interosseous stuff, but I think the Hernigoo studies are amazing, right? And then when I think back to graduate school and some of the translational studies we did, I think back to the Pat Smith study for shoulder rotator cuff repair and testing all these different patches for a long time on a very relevant animal model. And I think that's been kind of a guiding light for dermal patches. And that was with Matt Smith from the University of Missouri, shoulder surgeon. So I think that one, because part of graduate school, it was animal care. So it's like we definitely saw firsthand exactly what was working and what wasn't. And it was pretty dramatic in my mind. So I think those two types of things really showed me a lot of things and really was able to, I think, revolutionize orthopedic medicine.
All right, so lastly, Missouri's given us Mark Twain.
Yeah.
If Twain had to describe exosomes in a witty one-liner, what would he say?
Yeah, so I guess a little history on Mark Twain. That's not his real name. His real name is Samuel Clemens. So try not to bore you with too much history, but
you see what I'm talking about? He literally knows everything about Missouri.
Grew up there. I took a history of Missouri class. Fun story, I got kicked out of that class twice because I asked the teacher too many questions. She didn't like it, but that's for another day. Yeah. But his real name is Samuel Clemens. When he was an author, he was actually working on ferry boats up and down the Mississippi River, or Missouri River. And he was from the Hannibal area, and he was starting to begin his journey with being an author, and he didn't really want to go by Samuel Clemens. He didn't like the name too much, I guess. So he was trying to think of what can I call myself as an author. On these ferry ships, they had a term where they had to check to make sure their boat would make it through a certain portion of the river. 12 feet was their marker, right? And so I think it's 2 fathoms or something like that is 12 feet, and a shorter name for that is Twain. So when you're going through the river, they'd have their sticks with a mark on it, and if it goes past that mark, they'd yell out Mark Twain.
Yeah.
And so that's what he went by. But back to your question, I think he would maybe talk about himself a little bit and be like, I think we need kind of a Mark Twain in this area. We need something that kind of guides us and say this is safe and this is effective and we're going to be all right down that river and we're going to be all right treating our patients. So I think he might be a little witty with that.
Witty with that.
Yeah.
That's awesome. I mean, I don't know how you came up with that answer, but that's great. Well, thank you. This has been awesome. I hope you guys enjoyed this. Don't forget to hit like and subscribe to the channel if you enjoy this and more content. Preston, thank you so much for doing this.
This was a pleasure. This was so much fun.
Awesome.
Let's do it again sometime.
Appreciate it. All righty. See you guys next time.
Thank you.